Hello, everyone, and thank you for tuning in to another episode of Bugs, Drugs, and Genomes, where we discuss the incredible work ongoing at the DTU GlowCap Group. Today, we are joined by Jette, who is a food microbiologist, and Joao, who is a bioinformatician. We will discuss more about the daunting yet intriguing concept of biowarfare and how it relates to our work in the GlowCap Group. So, let's get started.
Hi, Jette. Welcome to the podcast. We are really happy to have you here with us today. Welcome again. And let's talk a little bit about your work in the Global Capacity Group. Before we get started with more specifics, could you please introduce yourself and tell us a little bit more about your background?
Thank you, Joao. Yes, of course. My name is Jette, and I'm a food microbiologist, and my background is in zoonotic pathogens like Salmonella, Campylobacter, and E. coli, which are all bacteria that can cause disease and which we get in contact with through animals or food. And in the GlowCap group, I work mainly on antibiotic resistance and the spread of this in both humans, foods, and animals, and the environment. But also, I'm part of several projects, including collaborations with the UNSGM to support the mission to strengthen laboratory participation in biological arms control and improve methodologies for genomic-based pathogen data analysis and also interpretation to detect alleged use of biological weapons.
Wow, that sounds really incredible and super interesting. Thank you very much for these insights. Since you mentioned it, can you please tell us a little bit more about the UNSGM? What is it? What the project is? What are more like the details about it?
Yes, of course. So the UNSGM is an international mechanism which can carry out investigations if there is a suspicion of alleged use of chemical or biological weapons between countries. So if a UN country is attacked with, for example, bioterror weapons, then there's this mechanisms that can go in place and start investigations. And so from a start, we help build a more solid list of laboratories in the UN that they can use for investigations. So they have this list of laboratories, but it's not really fulfilling, and they wanted to know more about the laboratories on the list and also how they can include more laboratories. So in collaboration with the Robert Koch Institute in Germany and the Swedish Defense Research Agency, we have been looking into this roster list of the UN and also on how we can determine the capacity of the laboratories here and also how we can include more laboratories on this list. So we have made genomic challenges for them to solve and to have an idea of which labs that can do this kind of work to investigate outbreaks. And we have worked both with bacteria and virus in this context. Recently, we have also been working with in-field sequencing to be able to identify pathogens directly on the site of an outbreak instead of shipping samples across borders, because that can be really problematic if you're working with an unknown agent and you want to move it across one international border to another country. So it would be more feasible maybe to do the sequencing right on site and do the bioinformatics analysis right on site and then go from there.
Thank you, Jette. That sounds really fascinating. Since you mentioned biowarfare, could you elaborate on why this topic is important? And especially for people who have no background in this area, how does the Global Capacity Group help in this mission of raising awareness?
Well, biowarfare is of course important because we need to have a preparedness in case some outbreak happens with maybe an unknown agent or like an unusual agent. And we want to figure, is it a natural outbreak or is it something that could maybe have done on purpose. So we need to have laboratories that can actually go in and investigate this and can take a decision. Is this something that is critical? Are we looking at some biowarfare, or is it just maybe an unusual but natural outbreak? And that is what we want laboratories to be able to distinguish between. So for that, we have been evaluating the capacity of laboratories using these proficiency testing schemes that we have been doing on both bacteria and virus, and to see how good are they at characterizing different bioterror-related agents. So bacteria and viruses that we know could be used for biowarfare purposes. So as part of these proficiency testing we have been doing, we always have some user cases also where the participants in the proficiency test also show how they have been doing. And this is also kind of a capacity building workshop, so other laboratories can also get inspired. And then we are working on also making more bioinformatics training also in relation to these biowarfare context.
So, since you mentioned these proficiency tests, can you elaborate a little bit more about these tests, what these really are?
Yes, of course. So we wanted to evaluate the capacity of laboratories, not only the laboratories who are already involved in the like bioterror preparedness, but also like what I would say normal microbiological labs, so food and veterinary labs and public health laboratories also, to get these involved also to raise awareness about what is this mechanism even and how they can also play a role in these kind of investigations if needed. So we wanted to cover as many countries as possible. So we were looking for all kinds of microbiological labs. And what we do is to send out some genomic material. So we don't ship any pathogenic strains or anything around the world. We just send them DNA sequences and then we ask them some questions. They have to analyze them and then they have to interpret what they find and then they have to give some responses back to us. And then we evaluate what they are sending back and then we kind of give them a scoring. And it's not like they are failing, but we are just ranking them more or less so that the UN Secretary General mechanism will know more about the capacity of these laboratories.
And since you are talking about these results and how you evaluate this scoring, can you give us like a simple example on the beginning to the end of like a workflow on how you do this and how after having the results on what the labs can really like have when they receive these results?
Well, it's not so much for the labs to, I mean, of course, they can learn something when they take part in this, but we were testing them in different ways and we were working with both virus and bacteria. We started out with bacteria and we did a little bit of a fun trial where we were kind of mixing up in silico DNA samples and we wanted them to evaluate, for example, a normal salmonella that was now suddenly carrying virulence genes of a salmonella typhi, for example, or for some completely different pathogen also. And then they had to find these pathogenic traits from other bacteria. And that was kind of interesting, but it was also very artificial because it was just something that we kind of made up in the bioinformatics. So that was one way of trying to see, can they find a threat in a normal data set? And then we also worked on different outbreak scenarios. So we were sending them also like metagenomic samples from foods where you have sequenced the entire community of the bacteria and viruses and whatever is in a food sample. And then they had to find again maybe an outbreak strain or a potential bioterror related pathogen or something in this data set. And we also did more outbreak related investigations where we sent them a lot of strains, like maybe hundreds of different strains, and then they had to find what is causing an outbreak. Is it related to humans or animals or the environment? And then they were looking into like small differences between the bacterial strains, and then we were evaluating how well they performed on this.
Also, about this project, how many laboratories are included in this project?
Yes, we wanted to include as many laboratories as possible, and we wanted to have as wide a coverage on a global scale as possible. So we were using our existing networks in Africa and South America and Asia also to include laboratories from countries which is not already on the roster list. So we were inviting laboratories to take part in this, even though they are not really very proficient, but maybe they are just beginning to do bioinformatics, and they also had a chance to show what they are doing here. And we actually had some really good results from like food and veterinary labs also. They perform well in this kind of testing because they have really good bioinformaticians. So even though they don't work in biopreparedness typically, they still have a chance to work in this field because of their good capacity.
So we were talking about sending DNA samples, but you also mentioned in-field sequencing. What is this in-field sequencing?
Yes. So this was like a technical test of what can we actually do in field. We have now these handhold sequences which you can bring wherever you have a laptop and some internet and some power. You can do sequencing. You don't have to be in a lab. So we sent out actually both DNA samples but also like inactivated bacterial samples, and then we asked the laboratories to do an on-site sequencing. So they were using only portable equipment. We had this small laptop-sized laboratorium where they could do certification and they could run a PCR and they had a heating block, so they could actually do all the lab work on that small laboratory that we sent to them also. And we sent them these DNA samples and also the bacterial samples, and then they were going to extract DNA just to compare their own extraction of DNA with our extraction. And then they had to sequence all the samples on this handheld device. So we were sending them bacterial samples, both some that we know are difficult to work with and some that are easy to work with. And then we wanted to compare. If we take these, we had 10 samples of bacteria. We sent them out to laboratories all over the world, in Asia, in Africa, in North and South America, and in Europe as well. And then we wanted to compare how reliable is the sequencing, how reproducible is the results that we get when we send out samples like this. Can we actually rely on the sequencing that we do in field, and can we use that for analytical purposes if we want to solve an outbreak this way?
So first, thank you very much for all this explanation that you are giving to us, and I'm sure our listeners will find this very insightful. And I have one more question for now. Why is DTU Food Institute involved in these bioterror defense collaborations?
Well, that was kind of a coincidence. So we have at DTU for decades been performing laboratory training and proficiency testing to improve the capacity of laboratories worldwide in antimicrobial susceptibility testing and whole genome sequencing and genomics. And as the reference laboratory for several global organizations like WHO and FAO, we have very good contacts in laboratories all around the world, and we also know a bit about their capacities as well. And then we got in contact with people related to this UNSGM mechanisms who wanted to increase their laboratory network and to do some technical evaluations of them. So it was kind of interesting for us to also move into this area and use our network for other purposes than what they usually work with.
That is a very nice way of building bridges between laboratory networks on a global scale, and super interesting as well. For students at DTU who wants to learn more and more, where can they start? So if they are interested in working on projects like UNSGM project, the project that we are talking about, what qualifications are useful?
Well, a great starting point is the GLOCAP website, where we share information about our projects, events, and resources. But students can also explore sources like the UNSGM website, where they can learn much more about these mechanisms, or they can look at the Danish Center for Biosecurity and Biopreparedness, the CPB. And then, of course, if they want to work in this field, it's important that they have skills in the microbiology or epidemiology or bioinformatics, which is what they are currently looking for. But they're also working on toxins and chemicals. So, I mean, the UNSGM work on a broad scale.
Thank you so much for joining us and giving us such an insightful look into your work. We are grateful to be a part of a group doing such meaningful and impactful work. Thank you so much, Jette.
Well, thank you for having me, Joao. It was a pleasure to be part of this discussion.
Thank you both for such an insightful conversation. This wraps up another episode on what we achieve at the GLOCAP group every day. Before we wrap up, we have a quick quiz for our listeners. The quiz question is: what is bioterrorism? Option A, the use of chemicals in warfare. Option B, the use of biological agents to harm or frighten people. And option C, the use of nuclear weapons. And the right answer is option B, the use of biological agents to harm or frighten people. Thank you all for listening, and we will see you in another episode of Bugs, Drugs, and Genomes. See you!